So welcome everyone to the Solanaceae seminars online. We are here meeting again every 2 weeks, starting the second season of the year. And today I'm glad to introduce María Laura Vidoz. She has a degree in agricultural engineering from the Universidad Nacional del Nordeste in Argentina. And she got her master's in the University of Florida and her PhD in the University of Pisa. Since she encountered tomato plants, she never stopped working with them, and she currently is a full professor in plant physiology and adjunct researcher in CONICET in Argentina. Today, she will talk about her passion in Solanaceae, tomato plants, and the responses to flooding stress. So with that, you can start sharing your screen and starting with the talk. Thank you, Maria, for being here today.
Okay, thank you for inviting me to present our work with flooded tomatoes. I'm going to start telling that I'm working in the northeastern part of Argentina, which is an area prone to flooding, not in the past few years, but it does get flooded when it rains heavily. And this is an important horticultural area also in Argentina. And I want to start talking about why we work with flooding stress in tomatoes. And as you all know, because you may have read about it or suffer its consequences directly, climate change has been threatening our planet, and it results in extreme events in terms of temperature and water availability. And here you can see flooding events in different continents, and it has indeed increased over the past decades. And tomatoes do get flooded, so it is a problem that they have to face. Then I want to talk why we have chosen to work with tomatoes, and not only because of its economic importance, which nobody can deny, but mainly because it's a great model species, not only for the study of fleshy fruit formation, but also for other physiological studies. And now that its genome has been sequenced, it's even easier to work with them. And in particular, we are interested in everything that has to do with primary roots, and more specifically also with adventitious roots. And tomatoes are great plants for that because they usually form adventitious root primordia along the stems as the plant gets older. And also because we believe that some basic studies about plants are necessary to then adopt the best and more sustainable management strategies. So we are in favor many times of basic science, also because it will lead to better applied science or management. So first we have to mention some of the threats that the excess of water implies. First is the hypoxia of the tissues that are underwater. The available oxygen doesn't last long, and so those tissues have to face hypoxia. Also, when the water level covers some leaves, there is a reduction in light incidence, which affects photosynthesis, and it's also affected by a lower carbon dioxide availability because gases have a much lower diffusion rate through water. Also, some nutrients may become more available, others less available. So that's another aspect that is affected in this stress. And as a consequence of the water that surrounds the plant, also there is some ethylene built up, which is the hormone that is most frequently associated with flooding and one of the most studied ones under this stress. So when the plant faces this scenario, most plants, those that can tolerate the flooding stress, have a number of acclimations. One of them is the formation of adventitious roots, because the original roots, those that were already present in the plant, in general die within a few days of being underwater. So the formation of a new root system is important for the subsequent growth of the plant. Then the formation of aerenchyma is essential for these new roots to receive enough oxygen to keep growing and functioning. Then in rice plants in particular, but it has been studied also in some others, in some legumes, there are two interesting strategies. One is called the escape strategy, and the other one is the quiescence strategy. What do they mean? In the escape strategy, the plants elongate very fast to regain contact with the atmosphere. And in that way, the leaves that are in contact with normal concentration of oxygen can diffuse to those leaves, and then through aerenchyma, oxygen reaches the tips of the roots, and so the plant doesn't suffer really from hypoxia. This strategy is an advantage when the water level isn't so high that the plant exhausts its reserves before it regains contact with the atmosphere. The other strategy, the quiescence strategy, refers to the plant reducing its metabolism to the maximum so that it preserves its reserves until the water level goes down. So this case is an advantage when the water level may be high or low, but it doesn't last for too long, because in that case, the plant would exhaust the reserves before the water level goes down. So they are an advantage in different conditions of flooding. So now, knowing this of the plants in general, let's go to see what happens with the tomato plants. And in our studies, we work in general with 4 week-old tomato plants, so in the vegetative phase, and the water level that we in general apply, unless I will specify, it's a partial submergence, so that only the base of the plant is underwater. In some cases, you can also study complete submergence, but that's lethal for the tomatoes. And waterlogging, which is when only the roots are affected. So since the base of the plants, and in particular the roots, are the first organs that have to face with this stress, we started studying to see what happened at that level. And in the first studies, we have here in the top, in this line, we have the control plants growing in control conditions, and below we have flooded plants, and with the dashed line, we indicated the level of the water. And you can see that after 72 hours of being underwater, the primordium, this stem, starts to elongate. And this phenomenon is very conspicuous after 7 days of flooding. When we did some anatomical studies, we could see that even after just 24 hours of being flooded, the primordia are already active and growing within the stem tissues. So this phenomenon is very fast. It starts with just a few hours of flooding. since the ethylene is the hormone that is usually associated with flooding, we wanted to know how it was affecting our tomato plants and also which other hormones were implicated. So in the first studies, we observed the expression level of some enzymes that lead to the formation of the ethylene precursor. And we saw that they were highly induced in submerged organs, which are the black dots. We measured ethylene formation in control and flooded plants, and we saw that flooded plants or even flooded organs such as leaves, the epicotyl and hypocotyls produced at least the double of ethylene. So to check on its role on the adventitious root formation, we used one of the many monogenic mutants that are available for tomato, which is another advantage using this species because you have many mutants. And it's a never ripe, which is less sensitive to ethylene. So we flooded the never ripe plants and we found that they produced a lower number of adventitious roots and they were also shorter. We measured ethylene formation under these conditions and we saw that never ripe plants are able to produce ethylene, but they just don't sense it. So up to this point, we knew that ethylene biosynthesis and signaling are needed for the formation of adventitious roots. Also, since an increased level in ethylene may be related to a higher content of auxin, for example, and knowing that auxins are responsible for root formation, we want to see the auxin level in wild type and in the never ripe mutant above and below the water level. And we saw that flooded plants that are seen here in black bars have a higher content of auxin in the wild type, but flooded plants of never ripe fail to increase their auxin level. And so we don't have this increased auxin and we see lower formation of adventitious roots. Now, we started then to explore this auxin, the auxin participation in this phenomenon. So we did a gas assay and saw that auxins were indeed in high quantities there. And we wanted to confirm that by using another mutant that is available, that is the Agiotropica mutant in tomatoes. That is a mutant with a very marked phenotype, and plants are in general like weaker stems, agravitropic, lower apical dominance, and as you can see here, even the few adventitious roots that form at the root neck are going upwards. So it's a mutant that is less sensitive to auxins. And as you can see here, the formation of adventitious roots is practically absent in most of the hypocotyl, just a few at the neck, in the neck of the plant. So up to this point, we knew that auxin accumulation there and perception was also needed for the formation of an abundant adventitious root system. When we went to see the ethylene production in these plants, we saw that in our wild type, there was a high formation, a higher production of ethylene as we expected, but the Agiotropica failed to produce a peak in ethylene. So also auxin perception was needed for this ethylene buildup. So we had these two hormones acting together to produce adventitious roots. And with this and some other studies, we could propose this scheme in which we say that flooding leads to ethylene entrapment, which increases what's oxygen transport towards the base of the plant, and through the DGT gene, it increases ethylene synthesis and perpetuates this cycle. This increase in oxygen transport towards the base of the plant also results in the formation of adventitious roots. So the next question is, because we have a plant that is producing a lot of adventitious roots underwater, so how important are they for this plant that is facing this stress? And here we used yet another tomato mutant that is called aerial roots, and it's characterized by the profuse presence of adventitious root primordia along the stem. These primordia just stay dormant until some external factor starts to act, as I will show you in a little bit. We observed that this aerial mutant starts to produce adventitious root primordia earlier during its development than the wild type. So when you went to count on the number of primordia, here you can see the difference between the air mutant and Ailsa Craig. While the number is quite low in Ailsa Craig after 4 weeks of growth, in the air mutant, there is a impressive number of adventitious root primordia above and below the hypocotyl, the cotyledonal node. So it was an interesting mutant to try under flooding, but there wasn't much information about it. So in order to use it, we started to characterize it first before we could say if it we could answer our question if it was if adventitious roots were an adaptive response. So the first thing that we did was the easiest one, that was to decapitate plants in the wild type and in the air mutant to see a variation in the number of primordia. And also we did another treatment with decapitated plants and application of auxins to see if we could restore the phenotype by adding IAA. So this would give us the idea that the auxins were responsible for this phenomenon. And indeed, we saw a reduction in the number of primordia in decapitated plants. So we removed the apex as a site of auxin production, but we could restore the original number by applying IAA. The same was observed when we used an inhibitor of auxin transport. And here you can see that by applying TIVA, you completely block the formation of adventitious roots. So we needed auxins that were formed in the apex, and we needed them to move to the base of the plant. We then wanted to know if this was due to a different sensitivity to auxins between the wild type and the mutant. But when we did 2 essays, one regarding hypocotyl elongation in increasing auxin concentrations, and also when we analyzed the ability of hypocotyls to root also with increasing auxin concentrations, we saw a similar behavior between both genotypes. So we concluded that it wasn't due to a differential sensitivity to auxins. Auxins have also something particular, which is their polar movement using some carriers that are present in the membrane. So we analyzed the content, the expression of these carriers in the wild type and in the air mutant. Here we have the PIN gene, the expression level, and here lags, which are an efflux carrier and an influx one. And we could see a differential expression between both genotypes. While in the wild type, the expression increases towards the base of the plant. In the AR mutant, their expression decreases towards the base. Also, when we analyzed the LAX5 gene. So, this gives us the idea that there is a faster transport in the mutant in the upper part of the plant, but it goes slower in the base. So if you have the transport that is very slow, we could expect an increase in auxin content. But to confirm first our idea that the transport was slower, we did another experiment in which what we did was to take a piece of the stem at different levels. Then we incubated it with label IAA. After the incubation period, we cut it in 2 parts. And then we measured how much of the auxin was in the upper and in the lower part. And in this graph, you can see how much was transported, the percentage that was transported to the lower half in each of these pieces, considering the apex, how much in an apical portion, how much was transported towards the base of that apical portion of the stem, and in the hypocotyl, how much was transported to the lower portion of the hypocotyl. So this movement of auxins in the apex moved very slow or slowly in the wild type, and it moved faster in the hypocotyl. Whereas in the AR mutant, we have a high movement, it moves very fast in the apex area, and it's slower than in the apex in the hypocotyl. So there were some difference in the transport between the 2 genotypes. When we went to measure how this led to a different auxin content, we saw that in the wild type, very little content of auxin was present in the lower part compared to the apex, but in the mutant, we can see that we have a lower content in the apex respect to the other parts of the stem. So these different trends that we are seeing led us to propose that the slower transport in the mutant, in the base of the plant, leads to an accumulation of auxin, and this results in the formation of adventitious root primordia. Now, knowing how it was caused, we then started to use this mutant in flooding experiments. And here you can see how impressive the formation of adventitious roots is, in particular in the AR mutant. We have 2, 3, and 4-week-old plants of our wild type, which is AC, and the mutant, which is aerial roots. You can see that even 2-week-old plants that are quite little produce a considerable number of adventitious roots, and it increases as the stem gets older, because here you have more primordials that are already formed. When we analyze what happens, how the formation of roots goes along the days, we can see that the AR mutant starts earlier to form these adventitious roots. So in the end, we could see, I picked 2 parameters that are very interesting. One is the total leaf area and the total biomass for control and flooded plants for 1, 2, and 3 weeks in the wild type and in the mutant. So if we look at the total leaf area, we can see that in the wild type, the flooded plants never reach the control level. But in the AR mutant, which you could see how many roots it forms, after 3 weeks of flooding, the level of total leaf area in the flooded plants was the same and in control plants. So it could keep growing, even underwater, with roots underwater. And also when we observed the biomass in the wild type, it was always lower than in control, in flooded plants than in the control ones, but flooded plants of air are able to reach the control levels after 3 weeks of flooding. So it keeps growing, keeps accumulating biomass in spite of being under this stress. So the air mutant has an advantage, forms adventitious roots faster, and it has an advantage over the other genotypes. And one of the most interesting things is when we analyze the root to shoot ratio. And here you can see that when you consider the original roots, in both cases, it goes down because the original roots die after a few days of flooding. But when you consider, you make the root to shoot ratio using adventitious roots, you can see something very interesting, that is that the air mutant has a similar level of root to shoot ratio as the control. So the new root system practically replaces the roots that die because of the flooding stress. Now, we have roots that are growing underwater, but they need to receive oxygen in order to keep working. So our next study was to see how oxygen was arriving to that area. And so we are now focusing mainly in the stems from which the adventitious root formed. The first study was regarding the porosity, and here you can see the porosity in control plants, in flooded plants in gray, that it increases after a few days of flooding, and in black you can see flooded plants that were treated with silver nitrate, which is an inhibitor of ethylene perception. So this graph not only gives us the idea that flooded plants increase their porosity, but also that ethylene was required. So we, of course, used our never-ripe mutant to confirm this, and we saw that in never-ripe there was no such increase in porosity. Also, when did some sections, we analyzed them, we could see that flooded plants, after 72 hours being exposed to flooding stress, they started forming lysigenous aerenchyma. And this wasn't seen when those stems were treated with the ethylene inhibitor. We then did some Evans blue staining, and here you can see in control, in flooded plants and flooded plants treating with the inhibitor, and we saw that flooded plants really have a large number of cells that are going to die. That is what this staining is telling us. And those cells are those that are going to disappear, to leave those huge spaces through which oxygen can diffuse to submerged tissues. And here below, you can see that this staining also marks a large number, an important number of cells that are about to die right outside the adventitious root primordia. And this is important. This is something that has been studied in rice, for example, because it loosens, having cells that die in the cortex, it loosens the tissue so that the meristem of the primordia don't die while they are elongating through the stems of the plant. This staining indicates the regions also where reactive oxygen species are formed, that they are also in sometimes like sensors and signaling molecules of the flooding stress. And we realized that both, also with the tunnel study, that programmed cell death together, induced by ethylene, together with reactive oxygen species, were acting together to form this aerenchyma in flooded stems of tomato. This phenomenon of aerenchyma formation is also accompanied by an increase in the diameter of the stem that is called hypertrophy. And this hypertrophy was present when stems were flooded, and it was reduced when we treated with the inhibitor of the ethylene. So once more, ethylene was involved in this increase in diameter. This was related to an increase in the cortex area in the flooded plants and an increase in the cell size. So we have a lower number of cells that remain alive, but these cells are larger. Then to check if the aerenchyma was actually working, we went on to analyze the internal oxygen content in control and flooded plants. And we saw that here you have the control with white circles. Flooded plants in which we inhibited ethylene perception, in which the internal oxygen content goes down. And here in gray, we have our flooded plants in which at the beginning of the flooding, the internal oxygen content does go down, but after 3 days, it starts to go up. And this coincides with the moment in which the aerenchyma starts to form. So everything was indicating that the aerenchyma was actually allowing oxygen to diffuse to the submerged tissues. So we continued our studies analyzing if this increase in oxygen content resulted actually in an increase in respiration. And we analyzed the respiration of roots and stems. And here you can see, in roots, we expected to see this, that is a reduction in respiration because the original roots die after a few days of flooding. But the interesting thing is that when you observe what happens with the stems, you can see an increase in respiration that goes above the control levels. And it happened also when the water level was like covering the stem, or the water level just covered the roots. It was just waterlogging in both cases. So this result was something that was hard for us to believe. So we started to confirm it with a number of experiments to see if it was actually happened that a stem that is below water has a higher respiration rate. And first we analyzed the ADH activity as an indicator of hypoxic environment. And we actually saw that submerged plants had a higher ADH activity at the beginning of the flooding. But once the aerenchyma is formed, it goes down. So the plant stops the fermentation. And what we did see was an increase in mitochondrial activity, which was analyzed through the content of formazan, the tetrasolium assay. So fermentation goes down and respiration goes up. We wanted to see if this oxygen consumption was due to the COX, the cytochrome oxidase activity, or if the alternative oxidase maybe was taking place. So we did another experiment in which under normal conditions, we had the activity of both enzymes. And then we inhibited the activity of the COX with potassium cyanide. So we had the alternative oxidase activity. And we saw that in that case, oxygen consumption almost stopped. So it was actually the cytochrome oxidase that was working there, consuming that oxygen. So we could, at this point, we could see and we could be sure that it was actually respiration, and those stems were consuming oxygen. It was also confirmed when we went to see the ATP content, and it was higher than in control plants. So it was a stem that was submerged, that was consuming oxygen, the respiration rate was very high, and it was producing ATP. So we said if there is a high respiration rate, then it needs some substrate. So we started studying the content of sugars in that stem. And here you have under partially submerged plants and in waterlogged plants. And you can see that the content of sugars is higher in either case. When water level goes up, the soil, or you only have saturated. So this increase in sugar content would provide a substrate for the high respiration rates. And the interesting thing here is that it doesn't matter if the stem is covered or not by water. In both cases, you can see it just needs the root to be affected to show an increase in sugar content in the hypocotyl and to see also an increase in the respiration rates. So to see where these sugars were coming from, we did a girdling experiment, and we could, as you can see here, when you have the girdle plants, you prevent the accumulation of sugars in the stem, in the base of the stem. This block in sugar transport toward the base of the plant also results, as you can see here in girdle plants, in a lower content, a lower consumption of oxygen and a lower mitochondrial activity. So if you don't get an accumulation of sugars, you also have a lower respiration rate. The sugars that are with an assay and the sugars that are being consumed there are the sucrose, and you can see an increase in respiration as the content of sucrose increases. But also the opposite happens, and you can see that when you block respiration, potassium cyanide, like in this case, you block respiration, but it also leads to a lower content of sugars. So both phenomena are related. You need sugars to increase respiration, but you also need respirations for sugars to keep coming towards the base of the plant. So basically what happens here is when you have a flooded plant, it forms aerenchyma, oxygen reaches the hypocotyl. When you restore the oxygen level, then respiration goes up and it results in sucrose accumulation. Also, the flooding affects the root because it blocks its functioning and it leads to its death. So with a dead root, the sucrose no longer goes there, and it is another factor that results in the accumulation at the base of the stem. Everything, all this leads to a higher respiration rate. So briefly, I will mention also what happens in the upper part of the plant when you flooded it. So I'm going to talk about leaves now, the part that is above the water level. We could see that in our never ripe, leaves stayed longer even when they were flooded. When we went to analyze chlorophyll content, we could see in gray you have the never ripe, that when it's flooded, it remains with the same chlorophyll content as in the control plants, but it goes down in flooded wild types. So a plant that keeps its chlorophyll A level gives you the idea that it's preserving its reaction centers and its photosynthesis may be also working under these conditions. That's what we had up to now. So we went to measure photosynthesis, the absorption rate, and the stomatal conductance, and we saw that both genotypes behave similarly, and you have no flow of carbon dioxide to the leaves, because stomata usually close within a few hours of being flooded. So we have stomatal limitations to photosynthesis here, but when we went to see the internal concentration of carbon dioxide, we saw that while the wild type had a normal concentration, it went down in never ripe. And it gives us the idea that the enzymatic part of the photosynthesis was working there because it was using the carbon, the internal carbon dioxide. We confirmed this when we analyzed what happened with increasing carbon dioxide concentrations, and we could see that flooded never ripe had a similar rate of CO2 fixation, but flooded wild type plants could no longer do that. So if you don't have CO2 fixation, you may think that there is a problem with the Rubisco. Therefore, we studied the content of Rubisco in both wild type and never ripe plants under control and flooding conditions. And here you have that never ripe, which is in gray bars, always have a higher content of Rubisco compared to the wild type. So the Rubisco levels stay higher even under flooding. Then the content of proteins is also higher in flooded never ripe. And the protease activity in never ripe is lower. So it's a plant that when you flood it, its leaves have a lower proteolytic activity. So we think that at this point, okay, then it's preserving the Rubisco. Also, the expression levels of some cysteine protease genes that are related and so associated to senescence are lower in the flooded never ripe compared to the wild type. So all these can make us show this little scheme in which a flooded plant that produces more ethylene leads to when you have high levels of ethylene, and as in our wild type, we have a lower leaf area, lower content of chlorophyll. All these leads to a reduction in light harvesting, which will affect photosynthesis. But also high ethylene levels are related to a lower content of carotenoids. I didn't show that, a question of time, but never ripe keeps higher level of carotenoids, which is good because it protects the photosystems, because it dissipates the excess of energy. So all this in a regular flooded plant leads to a reduction in photosynthesis. If we consider on the other way, the flooding leads to stomatal closure. This blocks carbon dioxide intake, and therefore we have a reduction in Rubisco activity. But also the Rubisco is blocked because we also have an increase in protease activity in normal flooded plants that will affect the Rubisco content. So all this results in a reduction in the photosynthesis, which isn't seen as such is in the never ripe mutant. It only has a stomatal limitation. So to conclude, we have that ethylene that is produced in large amounts in flooding stress. It's although it leads to some adaptive responses, when it keeps at high levels, it will affect the photosynthetic performance. The new root system is formed through interaction between ethylene and auxin. The original roots die because of hypoxia within a few days of flooding, but the aerenchyma that is formed in the stem and in the new roots allows for oxygen diffusion and for these new roots to keep working. Also, the arrival of oxygen to the stem results in a higher respiration rates, and it converts the stem like in a new sink. Now we don't have the roots, so this submerged stem becomes a new sink. Now, I'm not going to talk much about this, but we are also starting some experiments with eggplants because they are known to be more tolerant to flooding, and they are used in some areas, even here, as a rootstock for tomatoes. So after waiting for almost 5 months because customs agency here was blocking them to arrive to Corrientes, we got the seeds by the end of last year, and we multiplied them, and we have started some experiments. And we chose, in general, I think we will continue with 2 varieties, the EG203 and the Florida market, which is a very widespread one. And we did the first studies regarding leaf area, for example, and biomass accumulation. And we found some differences among them. One of the most interesting one is the leaf biomass, which is greatly reduced in the Florida market, but we have no differences with the control in the EG203. And that's what more or less what we are looking for, some contrasting varieties to keep on with the studies, because there is very little now, just a few studies regarding the biochemical part in the roots. And also when we studied some fluorescence parameters, we saw that flooded plants of EG203 were very similar to the control ones, but in Florida market, plants were suffering more, especially regarding the 2 parameters. One is related to the dissipation of energy, but because it seems that more energy is dissipated than what it is actually used for photochemical reactions. So with this, I'm going to finish. Here are some past, still some that are still members of the group. We have Federico that did the photosynthesis. It was his master's thesis, the photosynthesis work. Patricio is an extra one. He's working with cold stress in grasses, so he's an outlier. And we have some undergrads. Cesar is starting, he's doing his PhD in oxidative stress in flooded tomato plants. And Majo is the one in charge of the eggplant project as a master's thesis. So that's all.
Thank you very much, María Laura. That was a great presentation, a lot of information about this process that actually how it responds these tomato plants to flooding. So now we are open to questions. I don't know if you want to stop sharing the screen and
okay,
and turn on your camera.
Okay.
Okay, so we have a question of Stacy Smith. She is asking about, she's wondering if there is a trade-off where when the plant is triggered to make all those adventitious roots by flooding, is there a trade-off with above ground growth? Like, do the plants grow slower, stay shorter, something like that?
Well, I didn't show this part. When the plant is flooded, it's like after 2 weeks, you still have a slower growth of biomass, upper biomass. But after 3 weeks, like you have such a large number of adventitious roots that it sustains the growth, and you actually don't see it that's different with the control plants. We are studying now what happens when you remove the water and you do some healing techniques, so you cover the new adventitious roots with soil. In that case, we do see, it's kind of complicated because you see the plants are doing better, but they tend to accumulate a lot of root biomass, and you don't see an accumulation of much leaf and stem biomass. So it seems that the plants sends most of its photosynthesis to maintaining that root system. It does keep the leaf area, so leaves must be thinner, but you have more leaves, but the leaf biomass suffers a bit in that moment, and while in the first days after flooding, because the plant seems to sustain mainly the support of the root system, because the original ones, they stopped working. We saw that even earlier than 24 hours, they stopped conducting water. And after 3 days of flooding, they just die. Most of the root dies.
Okay, that's great. I know if Stacy, are you around if you have something to add or? Oh, yeah, she said, well, that's really cool. Thank you for sharing. And then we have a question from Deidra Jacobsen. She says, if I understood correctly, this will be the response to a major flooding event, correct? Would you expect the same type of response with repeated water lodging?
Yes, this is just the flooding of the partial submergence of the base of the plant. We see similar responses with water logging, except that you don't have such an abundant adventitious root system only at the base of the plant. It's the formation of adventitious roots is slower. But we are actually, we have some experiments set up to see that repeated events of water logging, for example, or repeated flooding events. Because we expect that a plant that has undergone a first stage, it also depends on the intensity of the process. If we have some partial submergence, we expect that a second event may cost the plant a lot of the reserves that are already little after the first one, because if you have a flooded plant, it produces a lot of roots, and then it reoxygenates all the adventitious root system. If you don't do the healing, all those will die. So the plant has to face an oxidative stress at that point, and it's a very stressed plant that you are exposing to another stress event. So we believe that they will behave worse with the second event of partial flooding. Now, with the water logging, it depends also on the length of the event. If it's just one day, the roots they stop working, but they don't die. So the plant is in a better shape to tolerate another one. So you have to consider the water level and the length of the event. In that recovery work that I was talking about, we are hoping to see some differences in repeated flooding events, but we haven't actually carried out those yet. Also, we are planning to do a mix of stresses, also with heat stress and water logging stress, to see if they can be primed with a little stress before, if they behave better with the second one, or better or worse, but we want to see what happens there.
That's great, thank you, Maria. And yes, and I was also wondering about that. If you have seen there is some effect on because there are some fungi association between the roots of the tomato and well, this different species of fungi. So if Is there some response, or how does it affect like the flooding process to these communities?
Yes, we haven't studied the relation with mycorrhizal activity or with endogenous bacteria, which is very interesting because the tomato has a lot of things going on there. We haven't studied that part. We only did some hoping a postdoc that is starting in this line regarding not the mycorrhizal, but the interaction of tomato plants with pathogens, because we saw that while the tomato plants is flooding, there is an increase in salicylic acid, and a decrease in jasmonic acid. And this increase in salicylic acid, we suppose that is a defense plants from pathogens. So we did a few initial experiments, and we found that some defense plants, some defense responses are active, like some defense genes and all that. So we are hoping to start working not with the mycorrhizae, but with pathogenical fungi. Pathogenic. So that would be the project of a postdoc that should start now in November if she finishes her PhD on time. So it should be, we should have some results in the combination of biotic and abiotic stress.
That's great. Yeah, thank you. Super interesting as well. I don't know if there is any other question for Maria. Pedro said that great answer, that it was exactly what she was wondering. Thanks. Well, if there is no other question, so we are finishing the seminar. Thank you very much again, María Laura. That was a great talk. And in 2 weeks, we are meeting again for a talk of Gustavo Hayden from Brazil. So see you soon, and we'll be publishing the abstracts and all information for the upcoming seminars in the website. Okay, bye.
Okay, bye. Thank you for inviting me.